My name is Martin Hemberg, and I'm a group leader working at the Sanger Institute. And my work is in computational biology, and in particular working on a single-cell RNA-seq. And we first started giving this talk, this course, was it 2016, I think? And now we've been running it several times, so hopefully it has improved somewhat, the course material. It has certainly evolved. The big difference this time is that we have a large number of new people teaching the course. So pretty much everyone who's not whose Twitter handle isn't listed there is a new teaching at this time. So I'm very excited about this because it means that it will have a better chance of sort of surviving, and because it's getting hard when the people who originally gave it moved to the other end of the world, as Davis did. Yes, I think that's about what I have to say for the introduction here. So this material that we're going to be going through, all of it is available on the website, and there is also a Docker image that you can download and install, and that will allow you to run all the exercises and so on, and it will install all the packages and everything that you need to do this course material. As you probably heard, this is all being video recorded. So if you don't want to have your face seen on the interweb, you should avoid this space here. Is there anything else I need to say about this chapter? Sorry? Pad? Oh, yeah. So you can take notes, and those will be shared with other people. And obviously, we will also benefit from them because we will realize what things we need to make clearer next time. Because, as I mentioned before, this material is sort of continuously updated or semi-continuously updated. Right. So we'll move straight to, and yeah, the format of this is that the way it's going to work is that we're going to be sort of saying a few things about a certain topic, and then there will be a number of exercises for you to work through, and we will sit around here and come and answer questions that you may have, and then maybe at the end we will have a little bit of a wrap-up about each section, and then we'll move on to the next chapter, and it will be sort of going on like that for about 2 days. And the goal is really that you should be able to, once you get your sort of your files off the sequencing machine, then you should be able to know what to do in order to get to some sort of meaningful biological analysis for the sort of the typical kinds of questions that people ask from the single-cell RNA-seq data. A little bit about the background. So you're basically all familiar with bulk RNA-seq. It is assumed it was a sort of technology that sort of started coming into maturity in the mid to late noughties. And it allows you to explore the transcriptome of a sort of large population of cells in a very quantitative manner and in an unbiased manner as well. So about 10 years ago, the the technologies improved so that people were able to isolate individual cells and extract the RNA and amplify it, and sort of reducing the amount of bias, and so that you were able to use these technologies and techniques to quantify individual cells. So, however, it took a while for this to take off, about 5 years before the sort of protocols were sort of good enough, and also the costs had gone down so that this could start becoming more widespread. But I think it's fair to say that in the last 5 years, there's been a true explosion in this field. And now you see virtually every paper that does transcriptomics use these technologies. So I think the typical experimental workflow is sort of shown here. What people typically do is that you start out from some sort of solid tissue or some sort of biological sample. You need to sort of dissociate your cells. You need to isolate them, and then you need to extract the RNA, and need to reverse transcribe it into cDNA. This needs to be amplified. And then this is built into a sequencing library that you can run through your standard next generation sequencing machine. And then you can get a profile of the transcriptome in each of the cells. And then you can use this to do all kinds of exciting stuff. A lot of these questions were inaccessible using bulk RNA-seq. So it has opened up a large amount of new possibilities for understanding sort of biological heterogeneity, since after all the cell is sort of the fundamental unit in biology. So one of the challenges, though, is of course the computational analysis of this data, which is sort of opens up a number of problems that weren't that some of them you had in bulk RNA-seq data, and some of them are new. So here is the sort of the typical workflow that we're going to be going through. So these first three steps here in yellow are sort of standard for sequencing. So this is stuff that you will do with any type of sequencing data that you'll have. The orange parts here are sort of more specific to single-cell RNA-seq, but it's things that you will want to do regardless of what you do further downstream with your data. And finally, the blue parts here are sort of more sort of biological analysis that are more specific to the question that you have in hand for your research. So what we'll be going through is a sort of, it's not a sort of automated workflow. It's a sort of, it's more or less the standard way that people do it. But we're actually making you sort of look at each step individually to understand what is going on. There are these days several packages available that can do a lot of these things that we're showing you automatically. So in many ways, they might be handier to use. But on the other hand, they will be a bit of a black box. So the advantage of doing it the way we're proposing is that you'll get a better understanding for exactly what is happening, and which might help you troubleshoot things. So I'm not going to talk very much. So I think perhaps the biggest challenge that we have in analyzing this data and computationally is something that's going to come up several times here, is something called dropouts, which is basically when you have a gene is present in a cell, but for various technical reasons, which we don't fully understand, is that's sorry. It doesn't show up in the experiment. So this is something that we'll return to many times. And so looking at the sort of the different experimental methods that are available, this is one of my favorite figures from a recent review from Sarah Teichmann, which really shows the truly exponential growth that you've had here from 2009 in the first paper, which sequenced 8 cells, to in 2017 when 10x Genomics published a data set which has more than 1 million cells. I think the current public record is 2 million cells. I'm sure that there are various servers out there where even larger data sets are sitting around waiting to be published. So here is a sort of partial list of some of the most sort of protocols that have been published and that are in use today. I think this is obviously not the topic of this course, and there are sort of several good reviews that you can read about this. I think what is important for you to learn about here in terms of the technology is that they are sort of different, and they have different strengths and different weaknesses, and which one is best depends really on the specific problem that you have in mind. I think sort of microwells and fluidimes are not very popular these days. The stuff that most people use are these droplet-based technologies. So either DropSeq, where this figure is taken from, or the method from 10x Genomics. So this is again not really the topic of this course, but I would like to point you to a couple of really nice papers, and I think there are even more studies available now. Some of them have come out very recently, where people benchmark these different protocols and different platforms using various different types of metrics. And so here's one plot from one of these papers. It's about 2 years old now. And so here they look at 5 of these different protocols, and they look at the same samples of cells, and they sequence them to the same depth, and then they compare the number of genes detected. Now, you can see here a very significant difference between the different protocols. So this suggests that sort of what you actually find is not only a function of the cells that you had, but also of the technique that you used to quantify them. And here is another plot taken from another paper from the Teichmann group, where they compare even more of these different sequencing protocols, and you can sort of see that your accuracy, which measures as the correlation, can be quite different, and also the sensitivity, which is a very important quantity, which really shows you how few molecules that you can detect, really varies from sort of something in the range from 1 to 10 molecules for the sort of the most sensitive protocols, whereas others require sort of thousands or tens of thousands of molecules. So I think with that, we will jump on to the next chapter and the next person. Yes.
Right. Paul, do you have to put it really close to my mouth, or like that? Is that fine? Yeah, brilliant, cool, thank you. I'll just drag that in here. So, yeah, apparently it's recorded, so we need good voice. Hi, I'm Sebastian. Sebastian Müller, working at the Plant Science Department as a bioinformatician just down the road here. You can call me Seb, and I'm going to talk you through the first more practical things, and when it comes to single-cell RNA-seq analysis. So that's basically taking you from the raw data you get off your machine. So once you run your experiment, you usually get your raw data, and I'll talk you through what you should do with it. And because this is a 2-day course and the material is meant to be a 5-day course, I'm just skimming through some of the bits that are quite similar to bulk RNA-seq, which you have as a prerequisite to this course. So most of you should be familiar with bulk RNA-seq, not everyone, but I'll just skim through it anyway, so you get like a bit of a sense of what's going on, what's important. And I'll bring out the differences from bulk RNA-seq and single RNA-seq. So usually, the first thing you do after you have sequenced is downloading your FASTQ file. So usually you get a thing called a FASTQ file, which is basically a list of your sequences that you get. And in section 3.3, I'll just skip quickly forward. There's a bit of an example of what FASTQ files look like. That's basically a file, a text file with 4 lines for each sequence. The first line is a header. The second line is the actual sequence, so that's the important bit. The third one is a plus for some reason, not informative. And the fourth line is the sequence. So basically, that is being repeated millions and millions and millions of times over and over again. So that's what you get. Right, let's go back, and the first thing is to check the quality of the FASTQ files. That's done using usually a program called FastQC, which is actually developed here in Cambridge and the Baker Institute by Simon Andrews, and it's been used for bulk RNA-seq. So usually the FASTQ files are virtually handled the same as single-cell RNA-seq. So you do the same thing. You have to be a bit wary about the interpret results, but the handling is the same. So that's how you do it. You basically install the program and you just fire FastQC. So that's all on the command line. Bioinformatics happens on the command line. You might have realized that by now. So that's how you work it out, and you basically just specify the FASTQ files, and then you get some shiny reports. I'll just skip really quickly to section 4, where there's an example of what you get. Maria is going to cover that again, and probably, and that's basically a signature plot that you should get after FASTQ. So what you're seeing here is on the y-axis the position of the base pair and the read. So for that specific experiment, we had about 125 base pair reads. And this one is looking at what the qualities, the average quality for each read is at a given position. And the good quality is, so that's expressed in a so-called thread score, which ranges between 0 and 40. And 40 being really good, there's almost no sequencing error on your machine. So basically, you want to look if your machine run has worked out all right. And basically, it tells you how good your sequences are. So if you are in the green, which is between, say, 30 and 40 thread score, then you're good. That's considered to be really good. And nowadays, that's what you get most of the time anyway. But it's worthwhile checking. And you usually get a bit of a petering off towards the end. That's to do with the sequencing technology. Illumina sequencing tends to get worse quality as you sequence. So that's quite normal. But if you like in the red, you should be concerned and double check if there's something wrong with your sequencing machine or whatever. So that's something you look at, and the same for bulk RNA-seq. And I'll invite you to read more details on your own time. That's just to skim through it. And just, once you've made sure all the sequences are okay, the next step is trimming your reads. So sequencing usually works by attaching adapters to your fragments. And sometimes they are part of the sequence itself. If the insert is not large enough or some other reasons, you get parts of sequence you don't want in your, in your reads. And you have to trim them off. That's same for bulk RNA sequencing. And there's a huge area of software that can do that. One is called Trim Galore. Again, developed in the Bagram Institute by a guy called Felix Krieger, who's actually sitting next to Simon Andrews. But there's others, one called Trimmomatic, then you got Cut Adapt, and so forth. So there's quite a few. But basically, they take your FASTQ file, as here. So they take your FASTQ files and they give you a new set of FASTQ files with the adapters removed. That's called trimming. So again, same as for bulk RNA-seq. It's probably worth mentioning. So at this point, the difference between bulk RNA-seq and single RNA-seq is, as again, I'm going really quickly, skipping to one here. One of the most important things you might be taking away from today is that figure. So that's to do with quality control. So I'll just skip ahead for that. So usually in bulk RNA-seq, nowadays you do a thing called paired end sequencing. So if you have a transcript fragment, which is indicated here as this error, you usually sequence from that end and that end. They sort of belong together. And that's what you get as paired end information. So you got two reads that belong together on the same transcript. In single RNA sequencing, you actually use the second read, so that's not part of the transcript anymore. You use that to include the cell barcode. So that's been used for another purpose, and that is to keep track on the origin of the transcript in which cell. Because you're dealing with multiple cells, you need to have the information which cell a transcript belongs to. And that's encoded in read number 2. Okay, so instead of a second transcript information, you got the barcode information. That's really important. That's usually sitting in the first couple of base pairs. In DropSeq, for example, that's usually 12 base pairs. That's followed by UMI. Maria is going to cover that, but that's really important. So, some people argue you don't need to trim that read because usually that read is longer than the barcode information is. So you don't really need to trim that, but you definitely have to trim read number 1. Okay, so yes, yeah, exactly. So yeah, and that's so basically for for example, for drop-seq, when you encapsulate a cell along with the bead is covered with the oligos that all have the same sequence, and every read that then attaches to that one will have the same barcode, which is then on read number 2. And so you know this read belongs to that cell which was encapsulated with that barcode. Yeah. So yeah. So you got the barcode information for every single read you have. Yeah. Okay, so the question is the limited number of barcodes in a run. So I'm going to cover that for demultiplexing. There's a misconception on what demultiplexing means. There's two fundamentally different things, and I think that's part of the confusion. You're referring to the first one I'm going to cover in a minute, yeah. So I'll bring that out in literally a minute. Actually, no. Yeah, let's do it now. Demultiplexing. So once you've trimmed, you usually want to demultiplex the samples. So that's basically, in single cell, that's usually referred to separating the cells out. So because all the reads are being merged together, you want to separate them out for the cells. So each cell has the reads assigned to them. And it's really important to contrast that to what demultiplexing means in sequencing. So about 10 years back, so when you run multiple samples on the same sequencing machine, for example, the NextSeq 500 or sequencing machines you run nowadays, you usually run more than one sample in a flow cell. So and in order to do that, you have a third read, a so-called index read in the sequencing facility, and that basically gives you the ability to tell apart the sequencing samples. And that has nothing to do with single cell. That's just having several samples run on the same flow cell. And the process to separate out the samples again is also called demultiplexing. But usually we don't have to do that. That's usually done on the sequencing facility side. So that's kind of like forgotten that this actually existed because we don't have to worry about that anymore. But that was the original meaning. And I think that's what you mean. Yeah. There's a maximum number of 96 samples you can multiplex on a flow cell. And to demultiplexing that is usually nothing you have to do anymore, but it's weird concerning about demultiplexing the cell information from the read. That kind of clear? Yeah. Okay. So how do you do demultiplexing? There are several ways. So there's not one way in single cell RNA sequencing. So usually you have you want to pool together all the reads that have the same barcode. And one way to do that is Perl scripts. They are available at the Hamburg Group webpage, so that's all in the material. And for example, in this example, I think we have Fluidigm sequencing data. So this one basically takes the FASTQ files and writes out a file for each cell. So I think this one has around 400 different cells in them. So you get 400 files, and each of the file is named after the barcode, and it has all the sequences belong to that barcode. You got basically a file for each cell. Another strategy that's used by a software called drop-seq tools is not writing out different files, because as Martin has mentioned, some of the experiments have 1 million of cells. You don't want to have 1 million of files. That's not feasible because you got file limitations. So another approach is to have that tag in your BAM file associated with your sequence. So you have everything in one file, but you keep that barcode information as a separate information as a tag. So that's usually the two common ways you handle demultiplexing. That all right? Good. Yeah.
So does this barcode help in?
If the barcode helps to distinguish sample switches.
Yeah.
So if you switch.
Some samples.
I don't think that would help for that. Yeah, I don't think that's going to help you with barcodes, because the sequence of the barcodes are arbitrary. So it doesn't really tell you anything about
What does this problem?
Does anyone comment?
When you send data to facility, cell barcode to identify because most sequencing cell platforms. Yeah, so most single cell platforms will use the same set of cell barcodes for every experiment. So I've had this happen with two of my samples got swapped and we didn't realize until we were looking at the actual expression of the cells and saw that, well, the sample is supposed to be neurons, but it's expressing heart cell markers. So obviously this is not our neuron data set, but the cell barcodes were the same across both experiments. I believe 10x libraries sometimes include a unique barcode for the sample. So in that case, you could use those, but it'll depend on what platform you're using, whether that's an option or not.
Thank you, Talua. That was Talua. Okay. Right. Okay. Next step is you got your sequences. You need to know. The next step is you need to know where they originate from. For RNA-seq, usually you want to know which gene sequence originated from, and we usually do a thing called mapping. Again, it's exactly the same for single cell as for bulk RNA sequencing. So there's you can use all the software that's available for bulk RNA sequencing. For example, STAR. STAR is a sort of like quite fashionable mapping program nowadays, but others are BWA, TopHat, and both, yeah, loads. Bowtie is a big one, and yeah. So I think we're gonna, yeah, that's also mentioned in section 4 in a way. So it's a bit repetitive, but that's how you run. So basically, again, you do give it the FASTQ files you have trimmed and demultiplexed, and stick it into the STAR program again on the command line, and you get back the positional information of that read where it comes from in either the transcriptome or the genome. That's up to you in a way to decide whether you map it to the genome or transcriptome. And but usually you get the position back, and that's saved in a so-called BAM file, which you should know. But if you don't know it, don't worry about that. That's basically just a file that contains all the information for each sequence. And it again, it's listed in file formats, BAM file. And along with this positional information, you got all sorts of other information for each sequence. For example, the mapping quality, the length of this match, the mismatches, the mismatch precision, and this sort of thing. So everything that's to do with mapping is saved in the BAM file. Yeah, so you can have a look at this at your own time. And sometimes it's a good idea to actually look at the BAM file, and that's how you do that. So it can be quite informative. If you've never looked at the BAM file, I'll suggest that you might do that this way. But you can do that at your own time again. And a new sort of format is called the CRAM format, which is basically the same as the BAM format, but more compressed. Right, so I'll just skip that bit. And the last, yeah, so again, on mapping, on top of this normal mapping programs where you get BAM files, there's also like a new sort of mapping strategy called pseudo-aligners. So, for example, programs like Callisto or Salmon or Express, they're the big ones. So they don't give you the positional information, they just count. They're a lot faster, but they don't really give you the positional information of where the matches come from. So if you just want to count the number of reads that come from a gene, that's probably a good way to go. But if you want to inspect the reads and see where they are sort of located, if they are on an intron border or in an intron or an exon or whatever, then I would suggest to use the BAM format and those programs that would use that, which take a lot longer. So that's an active field of research, and I think, yeah, I'll just pass over to Maria, who's going to take you through section number 4.
quite a
It's a bit of
section and-
repetition, yeah.
Yeah. So I will be able to run through it quite fast. I think Sebastian said most of the important things. I think it also depends on the amount of cells you have, like how fast you want to do it. But I would say that at least I have. Oh, I think most people would use star or salmon. In my experience, that's what I hear the most, people using it.
So if you do 10x, you'll generally be doing the mapping with Cell Ranger. Cell Ranger uses star mapping to a filtered transcriptome. That's what it's doing behind the scenes. Most people would use star. Some people will use Callisto if they're doing full transcript sequencing and they're not using Cell Ranger.
So I hope this is okay. Yeah, my name is Maria, and I'm not working as close as Sebastian. I'm more at the Genome campus in the Wellcome Trust. But yeah, I will go quite fast here through the construction of the expression matrix because Sebastian covered most of it, so I will just be like a quick reminder. So yes, we get our reads back, and I think the important thing is that, of course, we will do some QC to get to the starting point. It will be our expression matrix. That it will usually, in each row, you have the expression of each row will correspond to a gene, and in each column of your matrix will correspond to a cell. And of course, there you will have the expression of each gene in a specific cell. And like Sebastian talked about, when you get the output, the first thing you'll do is that you run FastQC files. And like he mentioned, he showed you already a picture of an example of the FastQC output. And here, like he said, you see that you have a very few base pairs that are not completely, they don't show the best quality, but you see the overall picture is good, so you can continue with this example. And of course, there are multiple QC steps along the single cell analysis, and through there, you might be able to reject some of the cells. So you get these reads, and the quality of the reads looks fine. So of course, you will have to align these reads to your reference. You're working with human cells, you will align it to the human reference genome or to the mouse, and so on. And I think what it's important is that we know also that compared to bulk RNA-seq and single-cell RNA-seq, it's in single-cell RNA-seq, sometimes it's common to add spike-ins to your sample. So of course, you will have to add the reference of those sequences when you're doing the alignment, so you're able to match everything. And so when you run the alignment, this is an example of what you get. And in the x-axis, you have the cell, so each cell is represented by a bar, and here on the y-axis you have the millions of reads. And what usually you expect is that most of the reads should be uniquely mapped to your transcripts of the reference that you're using, and you might see that some will also be multi-mapped, but you see that it's a very small fraction, and of course you'll also have some that will be unmapped, depending on the quality of your sequence. Then here we have some red arrows that shows that these initial cells, they almost have very low amount of reads. So of course you will have to discard those cells because they will not be meaningful and might even add noise to your own data. So it's an initial QC that you do. And you can also see here that some of the cells represented with the yellow arrows, they seem to show a higher percentage of unmapped reads. But at this stage, we don't know what is the cause of this and how much will they influence the data analysis. So we keep them. And at a later stage, you can always decide to discard them. Yes. So when you do the mapping QC, you can use different ways to measure the quality of your mapping, like including the amount of reads mapping to ribosomal RNA or transfer RNAs, or the proportion of uniquely mapping reads, like I show you in the plot. But also, like what it happens with many single-cell technologies is that they use like 10x technology, it sequences through the 3' end. So in that case, you will also use different methods to do the QC. And here it's an example that it's looking at the gene pod percentiles from the 5' end and the 3' end, and you can clearly see that coverage looks very nicely for the most of the cells, but then you have these 3 outliners here that would probably correspond to the initial 3 cells that we have here. So in this case, you would discard these cells. And then, of course, I think the most important thing in the end you want is to do the reads quantification. What does that actually represent with the expression of my transcript? And to do that, we use the unique molecular identifiers, the UMIs, that Sebastian showed you, that they're already added. They're random barcodes, usually with 4 to 10 base pairs. And they tend to be most ended to the 10 base pairs. So you increase the number of unique molecular identifiers you have because you have multiple transcripts in every cell. And these random barcodes are already added to your transcripts during the reverse transcription of the preparation of your samples. You have the cell barcode that it defines a cell. And then after the cell barcode, like I can show, you have this UMI. And this, what it will allow you to do, is to measure the quantity of each molecule of transcript. So, yes. And that's how you do the counting of the barcodes. Hopefully, ideally, you would have a UMI that is only unique to a specific molecule, and that you will be able to, depending on how many you have, how many copies of that UMI you have, and that transcript, you will see if it's highly expressed or lowly expressed. Yeah. The wrong ones. I personally never did it at this stage. I do it after, when I have the expression matrix, and I do some initial cell QCs, because usually more you work with your own cells, you get a better feeling what would be the average of genes on those cells. And when you plot the average of genes and you see how many genes you see per cell, you'll see that you have cells that have low fraction of genes and, for example, high amount of mitochondrial RNA, which will show you that probably these cells were very stressed, or maybe in apoptosis, already disrupting, and so you will discard these cells. So, yeah, personally, I never did it at the FastQ. I usually, but again, this is also done by Cell Ranger, and the cell, I usually just work with the Cell Ranger output, and the Cell Ranger software has its specific QC that usually people just running as it is, but I know it can be tweaked depending on what you're expecting.
What do
You mean if you have doublets?
Yeah, if you have,
Question.
If I have duplicates of the read.
So, like some
Yes, but I think that's what we would call the doublets, the cell doublets. Okay. And there are multiple ways depending. If you're using cells, and this is, so if you have cells from the same donor, unless you can see that you'll have, that the average of your cell population, let's say, would be around 3,000 genes, but then you see some cells that are suddenly around 5,000 or 6,000. I would think that there's a bigger chance that these cells are duplicates. So there are 2 cells within the same droplet. And in that case, I would make a cutoff. Like, say, cells that express over 5,000 genes, I don't want to have those. and I will discharge those because I'm not secure, or I'm not sure whether this cell is a singlet or a doublet. But if some cases, and that's what we've also experienced before, you can pull samples from different donors. And there are specific softwares used, they're also called for demultiplexing, that based on information like of the SNPs of each donor, you would be able to pinpoint exactly the donor of each cell. Depending on the quality of the sequence, it will easily say that maybe it's a singlet. Sometimes it says it's dubious and vicious, so he's not sure whether it's a singlet or a doublet. And sometimes if the quality of the sequence is good, he will also be able to identify doublets. And then he says, by the software, you will be able to see, okay, it's duplicates from donor A and donor B. How many cycles you run?
Yeah.
Yes, for sure.
Okay.
I mean, I personally only run 10x genomics, but one of the information we give to sequencing pipelines, for the DNA preparation and everything in the library preparation, they ask us how many cells we actually expected to have loaded. And depending on the numbers of cells that you have loaded, they will either do more or less PCR cycles. And of course, if the quality of your cells will not be good, and if you're expecting a lot of cells and they don't do that many runs of PCRs, you will get less reads. So in this case, I always think it's a matter of the experience of your own cells. And it's something, of course, there are very nice protocols from 10x and definitely from other technologies, but sometimes things, they just don't work at the first time. And whether it's fresh tissue or frozen tissue, or how long has it been waiting on ice, or how rough were you with the treatment of these cells? Have these cells been previously stimulated with something or not? I mean, that will definitely vary from situation to situation. I'm not sure.
Well,
I mean, in the sense what I was asking.
duplication that
Yeah.
really.
So
Well, that's the UI really.
Yes, I would think
Yeah.
so.
Yeah, I'll get back to that one.
Yes, because when, yeah. Yes, but sometimes you also have sequencing errors in the UMI. So I think also what it's like, it's still an open question, and there's a lot of like softwares being developed, and there's no like, of course, there's a consensus way of like counting the barcodes, but it's still an area in research, and definitely there's still room for improving.
Just really quickly on that UMI and duplication issue. I think that's a really big issue also in bulk RNA sequencing. So usually if you run more than, say, 10 PCR cycles, you get quite high number on PCR duplicates. And then on ASeq, bulk on ASeq, there's no way to tell apart duplicates that arise as a result from PCR and duplicates that arise as a just normal fragment duplication. There's no way you can tell them apart. There's some sort of saturation math you can do, but if you say we do, for example, viruses, and if you the sequence of rice, you get like the same sequence over and over again, and there's no point where you can tell apart. In single cell, so you basically assume if the sequence is the same, the UMI is the same, it's a PCR duplicate. That's you might have errors, but there are not that many. So I think it's a safe assumption. So that usually just collapse it into one. Yeah. So that's what. Yeah. There was a question over there. Well, if the question was if the read ends at the same position, then yeah, so that's basically the same sequence. So yeah, again, if that also has the same UMI, then you just discard the second one. If it doesn't have the same UMI, then you assume it's actually a duplicated fragment, which is a real signal. So that's something you shouldn't discard. In bulk RNA, again, you can't just tell those things apart. So you usually, yeah. Okay. The question that you.
Yes, correcting for errors. Yeah, exactly. And that's what it also says here, that the account for errors in UMIs remains still an active area of research. Of course, we do it the best way we can, but definitely there's still room for improvement. But it's always important, I think, to do these plots that we're showing here, the amplification rate of your every gene, where you have the log of the reads with the UMIs, and you see there is some sort of saturation in the system. But also down in the downstream analysis, and I think going when we'll do some cleaning up in the expression metrics, it's also important to take into account the total of genes you have per cells and the total of molecules. And that's what I was saying. When you know you have an average of genes in a specific cell, you'll be able to set up some cutoffs, at least to avoid to have some sort of duplications or cells that were also in some sort of distress, and you don't want to include those in your analysis.
Okay, yeah, so now it's time for hands-on. But first of all, I was just been made aware of maybe some sort of practical advice that didn't come out as nice as it could be. So all the things I've mentioned in section 3 and 4, such as trim galore, star mapping and all that, that's used using Linux, using a bash console. So basically, you have never seen that before. That's what it looks like. So it's basically the console, and that's usually what we use for pre-processing, because it's a lot faster if you instead of running it in R itself. So for example, if you type star, that's how you run star, and then obviously you have to specify all the FASTQ files and all that. But there's help files, and in the material it tells you how to actually run those things. So that's how you use the console. And yeah, so but the hands-on will not be, so we're not going to use that because we're on a 2-day course, not the 5-day course. In the 5-day course, you would be doing that. But now we're doing RStudio. So that's a bit that's after pre-processing, which I invite you to open. So basically, now, if you haven't opened RStudio, do it now. The way you do that is clicking on that R on the left-hand side on your screen. So like that. And if you have opened that, that should have taken you into, you should see into the course material folder. And you should see a list of files here. Is that right? Do you see that? No? But do you generally see that list of files here? Good. And then I invite you to open, yeah? Yeah. Yeah.
That's what it is.
Okay. So yeah, in the course material folder, if you're in there, just click on course files, which is on your home in your home directory. Okay. If you have any questions, feel free to ask any of our demonstrators. So yeah, you're already going around, so they will help you about technical details. You don't want to. So okay. So and I'm now going through. Yeah. an RMD file, a markdown file, and I'll invite you to open an RMD file called expression QC. So that should be this guy here, exprs-qc.rmd. You click on that. That should open up that pane in here. That sort of okay? That work? Yeah, brilliant. So that's a markdown file. Just a show of hands. Who's familiar with R markdown? So that is just about half. Okay, brilliant. That's good. So just for the other half, just really quickly, R markdown is basically a mixture of R code with normal text. And usually the normal text is in markdown, so that's basically some sort of formatting of text. So for example, those hashes are indicating titles. Normal text just indicates normal text. And that's interspersed with R code, which, for example, is on line 26. That's usually marked by those 3 tick marks and a curly bracket. And everything between those 3 tick marks, they always go together, that's R code. And you can run that R code. So that one is not R code, and you don't run that, but you can run the things in between those 3 tick marks. And the way you do that is to click into that chunk, that R code chunk, and click on that arrow next to it. Maybe you can try that. And oh, that should, my console's gone. Oh yeah, so that should have copied that code into the R console and executed it. Did that work okay? Okay, so basically I'm now going through that section, and you're welcome to execute the code as we go along. So, so just for you to try it out yourself. Could you go on just to see if someone is struggling with? Right. Okay. Okay, I'll give you some time to set up. First of all, before I skip to the hands-on and to running code, just one of the sections we are skipping today is Introduction to R and Bioconductor, because it's prerequisite and we don't have time to cover that. But I'll just want to point out one subsection, so that's section 5. Subsection 5.7 talks about single-cell packages in Bioconductor. So I don't have time to explain Bioconductor, but that's basically R packages are specific to single-cell. And one is called single-cell experiment. That's a package in Bioconductor. Another one is called Skater. And basically, they are at the heart at what we're doing right now. So basically, we're running code that is provided by those packages. And this section, you can read it at your own time, gives you a bit of an introduction to those packages. And that's, can you actually read that? Just increase the font a bit. Where's it gone? Yeah. Yeah, so that is clunky. So for example, that is single-cell experiment is basically giving you a class, an R object, where you store all your single-cell information in. So that's quite central to what we're going to do now. And that's how you create such a class. So you basically provide it with sequence information, the read counts, and some metadata. That's how you do it. We do it like just live in a minute, but that's the general overview. And Skater is then basically doing math and some sort of all the things you want to do to analyze your data based on that single cell class. So that's doing stuff on that object. You're going to see in a minute. So you can do another sort of QC, data format standardization, normalization, visualization, and differential expression, and everything you could possibly dream of to do with your single cell data. Okay. So let's go to this bit. So what you just opened now corresponds to section 7.1 here. So cleaning the expression matrix. So an R, is that right? Okay. So that's, yeah, expression-qc.rmd in both files. Good. Yeah, just raise your hand if you can't find it, so someone will come and help you out. Okay, so introduction. So what we've just learned is to go from your FASTQ file into an expression matrix. That's going through trimming, quality control, and demultiplexing. And demultiplexing basically tells you which read corresponds to which cell, and mapping tells you which read corresponds to which gene. We end up using a gene expression matrix. Okay, and as Maria told you, the columns are the cell, and the rows are the genes. And I'll show you an example because they're always good. So that's what an expression matrix looks like. So the rows are individual genes. So that's just like a genome that might be 0 set or whatever. So those are usually about 30,000 of them. That's how many genes a human genome has, or whatever species you're working with. I care about plants. There's more genes, so the longer, more rows. So and the columns are the cells. And you can see here that's the number of reads that are included in the cell that are mapping to that gene. So that cell has 3 reads that originate from that gene. Okay, that's quite central to what we're going to do. So that's important to understand. And that's also something you get for bulk RNA sequencing. In bulk RNA sequencing, you usually have a few columns where you have, say, if you run an experiment with, say, 3 samples wild type and 3 samples mutant, you usually have a column for each. So you would have 6 columns, 3 wild type, 3 mutant, and you usually try to find genes that have a different count number between the 2 conditions. In single cell, the columns are quite a few more because you got a column for each individual cell. That's a lot more. And so you got up to, say, 1 million of columns. So that's really important. You got a lot more columns because each cell has a column. Also important to note is the numbers. So there's a lot of zeros. In single cell, because you have less sequences per cell, you have an average less reads overall. So you have loads of zeros. A lot of genes have zeros in it, and that's a really sparse. It's called a sparse matrix. And sometimes you have 1 or 2 or 3, but you don't have like hundreds usually. So usually you have like really low numbers, and that dramatically changes the way you analyze the data. That's the whole point of this course. The gene dropout issue.
Yeah, so that's a lot of zeros is what gene.
Well, it's it's not. No, it's usually just a consequence of low coverage, because you only have, say, 100,000 reads per cell. So you only have an average like 3 counts per gene, and some are more expressed than others. So if you have a 0, it doesn't mean the gene is not expressed. It might mean you just don't have enough coverage to detect it. So dropout is also playing into that, but it's mostly coverage.
You were mentioning about the read belonging to that gene, or the count that belongs to position specific to that gene.
So the question is if the read count the position belongs to that gene.
Because idea is like you not have many long reads, so some position.
Yeah.
So that's a count.
Yeah, that's a good question. So basically, how do you know when read overlaps the gene, and where do you set the boundaries? So that's handled differently, but the consensus is if there's even like a 1 base pair overlap with a gene, you just attribute it to that gene. Yeah, but it usually doesn't usually affect the analysis too much. So usually, if there's like a bit of overlap, you just attribute it to that gene. That's but you can change it. People play around with it, and they don't really find much difference. An intron, yeah, that's a good point. Especially if you look at nuclei, there's a debate on whether you should include intronic reads to your gene or not. And I think the argument goes that you should, especially if you look at trunk-seq. So that's just looking at nuclei, because you got a lot of preliminary non-spliced variants that are actually reads. So yes, usually people include the introns as well. Okay, good. All right, so that's asking about gene versus transcript, which is a big issue in bioinformatics. So do you count individual genes, or do you count each isoform of the gene separately? So because one gene might have different isoforms, that's what you're probably asking, which usually change by just like a bit, one or more exon, this sort of thing. And in this particular case, that's all isoforms together. So you don't distinguish between isoforms. And usually that's what people do, because only if you care about splicing and that sort of thing, you want to look at isoforms itself. And it's kind of giving it a whole new twist on things, because you run into multi-map problems, because most of the isoforms are shared, so you don't know where to attribute it to. So that's an active area of research. So we just, to keep it simple, just use genes rather than isoforms. Okay, so our studio, back to that. So the first chunk here is basically setting up the stage, and the second chunk here will load the mentioned libraries. So you just load in the libraries that makes all the functions that we need available. So, and the next one is reading in that expression matrix I was talking about. So that's how you do it. Boom. I can't open Classic. Yes. In order to do that, if you get that error, then you're probably in the wrong folder. So you need to be in the course material folder. If you try that yourself, so boom. No, hang on. Oh. Okay, so you have to just a second. Tools. How do you change the folder on that one? Yeah. So set working directory, open, and that should now work. Why is that? Okay, so we are data. Does that work for you guys? Yeah. Yeah, maybe we just do, hang on. Okay, it's a bit of R debugging. If you type in get WD, it tells you where you are located at the minute. Okay, and then that should work. Maria, do you have an idea why that doesn't? We just had it work like a second ago.
Because of the path? Yes.
Yeah.
How did we do that?
Yeah, we went tools. I don't know, was it tools? Session, yeah, and set working. But we are on the right one. That's course materials.
Oh, and it's not right?
Home participants. Oh, hang on. Sessions. Oh, it's the wrong one. So that is desktop. It's desktop course materials. I think that's how it. Okay. I was on the wrong folder. So it's not in home course materials. If you get the same error, it's not in home course materials, it's in desktop course materials. So I'll just talk you through it. So that's in sessions, and then set working directory, and then choose directory. And then you go on the desktop and select course material and open. That takes you in the right folder. Sorry. Oh. What? Okay. Does that work? No? No? Okay.
We should just rewrite the path. Yeah.
Yeah. Okay. Also, if you white that out, that should also give you. If it still doesn't work, then if you white that out, that should take you into the right folder. Because there's two course material. There's one in home folder and one desktop. The one desktop is there's the right one. Yeah. So just write that one out here, and then that should take you in the right folder. Is that working? Okay. So you just have to write it in the console. That's how. Okay, good. Ah, okay. I see. Good. Brilliant. Yeah. Yeah. That's probably a good idea. Yeah. For next time. That's a good note for next time. Good. Thank you. Okay. So we all good. Okay, and then okay, we just go on, and if you still have questions, just ask one of the instructors. But I think it works for most people. So the first thing, so what? Okay, guys, listen up. There's going to be a tea break in 50 minutes anyway, so I'll just bear with me for like another 50 minutes, and we just go through some of the files. So basically, what we did here is to load in the expression matrix, and that. And the second line is loading in the annotation, because the annotation gives you the information on which cell belongs to which experiment. Because usually also in a single cell experiment, you have cells that belong to several samples. So for example, if you want to compare tissues, you need to know which cell belongs to which tissue. And you have to specify the information yourself. That's usually going into an annotation file. And in this case, that's provided. We provide that for you, and you can inspect what it looks like. So that's in this chunk here. Okay. How is that? Okay, sorry. I'll just do that. Okay. Really? How did you? Yeah.
So if you ran get working directory.
Yeah. That's terrible. Okay. So if you have any questions on this guy, he knows all about R Markdown. Good, thank you. So that should work now. Okay, so that's what annotation looks like. So basically, that gives you a list of the cells that you recognize from the expression matrix and tells you which cell, which experiment does the cell belong to. So that's giving you the experiment information. So you need those two information to make sense of the experiment. Okay, and the next step, as I told you, is to bring that information into a single cell experiment class. So that's provided by that library I was talking about a minute ago. And the way you do that is using this sort of construct. So that's basically taking the molecules expression matrix and puts it into a slot in that object called assays. And the annotation goes into the cold data object. That's the way this class works. That's just like a given structure, and you just have to comply with it. And that should now work. Brilliant. And if you can inspect that by just firing up UMI. So I'll just increase that. Okay. So now we've saved that in an object called UMI. And if you look at it by just typing that out, it tells you a few informations. For example, the dimension of our expression matrix is 19,000 something and 864. So that basically means we got 19,000 rows, which correspond to genes. That's the number of genes. And that's the number of columns, which correspond to each individual cell. So we got 864 cells in our experiment. Okay, and that's the row names. That's the number. That's the genes. And that's the names of the cells. Okay, so that's all now contained in that object that we wanted to be in. Right, and Really good question. I think it's one sample. Talua, do you know about that experiment? So, well, but does it tell you how you run? I mean, yeah. Okay. Thank you, Talua. So the way you can inspect that, so just following up on that question, if you want to know how many individuals are there, you can use the table command and using the individual column in that annotation file. So basically, that tells you there's 288 of each for that. So there's 3 individuals with 288 members each. That makes sense? Yeah, good. And likewise, if you want, as Talua mentioned, the batch contains just about 10, I guess, different batches with 96 cells in each. Okay, so that's in this column here. Yeah, is that 10 different experiments then? Because that's 96 well plates, so there's literally 96 well plates. Thank you very much. Yeah, that's a Fluidigm C1 design. That's going to be different for, say, drop-seq. So you really need to know your experiment, and Talua knows. So thank you very much. Okay, so what is different to single-cell RNA-seq is the filtering. So as I mentioned, there's lots of zeros in this matrix, and we need to clean that. That's why this chapter is called Cleaning the Expression Matrix. Oh, yeah, by the way, that data set is actually published by those guys, Tang, 2017. And yeah, there's a few information in here, and so that's using a Fluidigm C1 platform. But in general, that workflow is valid for all other single-cell experiments and technologies. Also worth noting, that Skater package was written in Cambridge as well, at the EBI, I believe. Talua, are you part of the? Davis, yeah. So someone actually participated on that. And so is yeah, FastQC as well as TrimGalore. And to add to that, there's also a company, a Cambridge-based company that actually does single-cell machines called Dolomite Bio. They actually had some material next to the coffee machine if you are interested in that. So there's a lot of Cambridge-based single-cell things going on. Okay, so basically, to clean the expression matrix, we need to prune it. We need to make it more handleable. And usually, we just chuck out all the genes that are not expressed in any of the cells, and because they don't have any information. That's how you do that. You basically just filter out all the ones that have. You just keep the ones that have more than 0 cells. That's how you do that. Okay, you have to do that on yourself with a different sort of data anyway. So that's just a very logical thing. And another quite important step in single cell is ERC controls. So basically, that depends on your experimental setup. You can inject your RNA with a control RNA. So on top of your cell RNA, you can inject so-called ERCC transcripts. So that's a defined amount of transcript that you can use to gauge the amount of the transcript in your cell. So you can compare to your gene expression. And those ERCC cells, usually you have to incorporate it into your genome, and they are usually marked as genes. They're rows by themselves, but the name is not a gene name. It's got ERCC in its name usually, and you can identify it using a grep-l command. That basically tells you the rows that correspond to those spike-in controls. So those are not part of your cell. Those are control spike-in on A. That's how you do that. You have to mark them off as such. And the same for mitochondrial. So mitochondria have a specific meaning in single cell, because usually people have found if the mitochondrial contribution is quite high, you are likely to have a dead cell. So sometimes, for example, in droplet-based experiments, if the cell dies, for some reason the mitochondrial fraction goes up. Or sometimes you have ambient transcript. So say you have a droplet, and usually you have a bead and a cell in that droplet. But if you only have a bead, it will still sequester some of the ambient RNAs that are floating around in the experiment, and they are usually high in mitochondrial fraction. That's a telltale signal, tell a good from a bad cell. So you have to separate out the real cells from background noise. Okay, mitochondria might help you that. And that's basically a list of all the genes that are located on the mitochondria. So that's just an information you give it, and then Skater is going to work out that information, how much mitochondria there is. We'll see some plots in a minute. So that's how you do that. Sorry. Yeah. Question.
You know the ERCC?
Yeah.
Put it at.
Yeah.
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 So when you are actually.
Yeah.
You do it at
Before sequencing.
before sequence.
Yeah. So you sequence this. Yeah.
Yeah.
That's part of the sequences in your experiment. Yeah.
Oh, okay. Can't have it.
Well, yeah, you
So
wouldn't.
in the sense, I got a.
Yeah.
I'm not sure that could done in that.
Yeah. So the question was, yeah, if the ERC is being put before sequencing, and the answer is yes. Good. Okay, so that's marking those genes off. And now that function, so that's just a given. That function calculates, basically takes those genes in and takes that information in. So we've just marked off those genes by ourselves, and now we need to incorporate that information into that object. That's a bit tedious and boring, but in a minute you'll see some nice plots, and all the effort is worth it, I suppose. So okay, so we've got all the quality information and all the count information in our object, so that's all been saved in that UMI object. So if you remember, we have created the UMI object earlier on, and now we're just adding information into it. So we're just adding information about mitochondria, ERCC, and our experimental design and all that. So that's all completed now. So all the information is in our UMI object. And so what can we do with that UMI object? So the first thing is to look at the number of counts per cell. So that's usually what people are quite keen to look at early on. So you want to know how many reads are assigned to my cells, and you can use a histogram to do that. Okay, so that's basically in UMI. There's a column called total counts, and that gives you the number of counts that are associated with the barcodes. The cell is basically represented by a barcode, and that's the number of counts per barcode. And as you know, we have about 800 something barcodes, and each of them has a number of counts associated with them, and that's the histogram on how many those are. So we got about 50,000 to 100,000 counts per barcode on average. That's the bulk of our experiment. Some of them even have 200,000 counts to them. So that's really cells with like a high amount, and that's kind of reflecting biological variability. You have quite a range of reads associated with the cell. There's a question there. Say it again. Yeah, I'll get to that in a minute. So you're asking about that red line. The other end. Usually you keep that, yeah. But you might exclude them later on if they show some weird behavior. If they sort of are all right, you look at them in a bit more detail, but at this stage you don't exclude them. What you do exclude is usually the left-hand side. And I'll just cover that in 2 minutes. We'll carry on after the tea break in 2 minutes, but a really important thing in single-cell sequencing is the so-called knee plot. I'll show you what that means. That's actually in, whereas it's for some reason it's in demultiplexing. But importantly, if you run a single-cell sequencing experiment, no matter whether it's a Fluidigm or Droplet or 10x, you usually sometimes have beads that do not have contact with a cell, but they still have reads associated with them, but there's a lot less reads. You have the beads that are in contact with a cell, and you have the beads that are not in contact with a cell. Both of them are going to have reads associated with them. But you're only interested in the ones that have been in contact in the first place. How do you tell? Usually, the ones that have been in contact with a cell have more reads with them. So that's something we take advantage of, and that is something which goes in line with that histogram. You can rank the cells according to how many reads you have associated with them. And that's a typical plot. So that's kind of the same information as this histogram, but that's ordered. So that's basically on the x-axis, you have individual barcodes, and that's the UMI associated with a barcode. And that's a declining function. And here you can see you have a bit of a range, but here you see there's a bit of a drop, an inflection point. And that, in theory, should tell you the boundary between the real cells and the ones that are background noise. So that's considered background noise because you got a bit of a decline. That's considered to be your actual cells. That's how you tell them apart. That's quite important because usually you just throw them out. Okay, and there's so usually by just looking at them, you can say, "I'll just throw out those ones." But there's some mathematical tricks you can do. So for example, you can look for that inflection point yourself. There's some mathematical functions where you look at the incline and local maxima and this sort of thing. You just find it automatically. That's what usually people do. But it's kind of important to look at this plot and convince yourself because math sometimes goes wrong. And another approach is fitting distributions to your count read data. So that's basically a distribution for your cells and a distribution for your background noise, which is the green one. And you then basically use that mixture model to distinguish between the two. But as you can see, the ones that are supposed to be cells actually are bimodal, so they don't really fit to this one here. So that's probably not the best approach for that sort of data, but it might work for others. And the third one I was going to mention is actually, again, by a Cambridge-based lab, the Marioni lab in EBI. And they have a different approach. So they're basically looking at the background noise. So they're just taking the 10% most to the right as background noise and build a profile. So they're taking it one step further. So they're not only looking at the reads itself, the read counts itself, they're looking at how they're distributed across the transcriptome. So what sort of genes do they hit? And by building a profile based on the 10% that are very likely to be background noise. And then they build a metric to compare all the other stamps or cells, how similar they are to the background ones in terms of profile. That's how they decide. So there's a threshold. If they're like 99% similar to the background noise, they're chucking it away. So that's a more sophisticated approach. Did I get that right? Yeah, I think how it works. Okay. Right. So we have now. We're going to continue at this point in about 15 minutes time. There's going to be a tea break until 11:15. So just get yourself coffee, and then we'll get back to that one in a minute. Thank you very much.
You know where the knee is?
Yeah.
Yeah.
Is that?
Let's see if we
Same page. Yeah.
So, physical interpretation, is it that we thought there are 8,000 cells?
Yeah.
Because 8,000 barcodes, but actually, let's say maybe 2,000, let's say 3,000 barcodes actually managed to get attached to a cell. So actually, only, and the rest didn't really attach
Yeah.
to cells. So
Yeah.
basically, initially we were thinking there are 8,000 cells that have been single sequence, but actually it's only 3,000 which in reality have been single sequence.
Exactly. That's exactly what it means. Yeah. Yeah. Yeah.
So, okay, that's good then. And I mean,
And that's what the normal, that's how we think of it anyway. So there might be a chance that this one actually includes something that was in contact with a cell, but for some reason the amount of reads is really, really low. So it kind of like got confused with background noise. And that's why the Maroney lab came in, that like maybe we should go a step further, not only looking at the number of reads associated with the cell, but also the distribution on the cell.
Okay, so different ways of basically finding out if the barcode did manage, if the bead did manage to. And one of the things, like you were talking about that control, introducing control. So I got a cell sequencing.
Yeah.
It's from cancer human.
Yeah.
And now I don't know if the control was done, but I'll ask. It was in Germany, but I don't think it was done. But I can't do anything about it. But the second mitochondrial thing, is that something I can test if there are the cells which have got sort of they are dead cells or not?
Yep. So we're getting to that stage in a minute.
Okay.
Yeah. So that's something we covered. That looking at how to identify cells that are weird. Yeah.
So and that can be done with my data as well. In the sense, what I'm saying is, the control when I before sequence. There's no way I could do that. But mitochondrial will show up, isn't it?
Well, that can tell you which cells are
Dead cells.
dead cells. So I don't know. Yeah.
Okay, but the data I have got actually BAM files and fast. So there are 5 samples. So I got. I'm working on 3 FASTQ files. So each FASTQ file belongs to one cell basically.
Yeah.
So I think demultiplexing has been.
Yeah.
I got the data. And when I make the plot for the and my aim is to find out the heterogeneity between cells, so which are expressed one tumor and not the other,
Yep.
or the other way around. And I'll show you the matrix that in an Excel format. Surprised to see like there was hardly any
genes that are different between.
Actually, there are genes, but when I ran the experiment first time, no, yeah. So originally, the BAM files were for HT37. They were referenced to the human version 30.
Yep.
And when the reference I had was HT, the annotations I had
Yep.
used. So initially, it was not showing me any gene. The free cells have not got any gene at all because I was using the wrong reference.
Okay.
Yeah. So I sorted that problem out by mapping it to the HT37 now, and I do get some genes that are part of the. I'll show the expression matrix in like when you have time. But the other thing I wanted to ask was, so all the analysis they have done until now, it's by mapping it to HT37. Human genome, I think it's 18 or 19 version. 18.
Yeah, I'm plant scientist, so
Oh, you're
yeah.
plant. Okay, no, that's fine.
Not.
I mean, that's fine. So, but the real question was there are 2 different versions. One is 37 and one is 38. I think 38 were annotations, the reference.
Might have changed, yeah. I don't know.
Yeah, okay.
Ask someone, yeah.
I'll show you, because so you work on plant genomics. Okay.
Yeah.
So they're also like you're doing a lot of singles.
No, not really. Yeah, no. It's not easy for plants. Yeah, so that's kind of like quite new. So yeah. So I'll get back to you. Any feedback? Did I miss out on something? Yeah. Okay. Time wise. But you need.
No. While we are talking, they don't know our conversation.
Yeah. Okay.
No, I think it's I ask people and they were fine and they.
Okay. Yeah. Good. Brilliant. Thank you. Yeah. That's. Thank you very much. Yeah. That's good. Bit of feedback because so what you had in mind. Yeah. So that's what you had in mind and how going through that. And good, brilliant. Thank you very much. Any feedback, brilliant. Yeah.
Well, people are following it, and that's why I'm walking around. Because then you see, unless you see what people are typing, you have no idea from there. You don't know if they are lost or they are Facebooking
Yeah.
or they are like, oh my lord, and all of them were following
Yeah.
the same
Facing out.
No,
Yeah.
no, it was fine.
So they.
A couple are skipped.
That's why we get that, but it's like the majority of them following him as they
Oh, right. Good. Oh, brilliant. Oh, thank you very much. Cool. Good.
That's why we have all the materials there.
Yeah. No, that's really well crafted material.
always to go ahead, fine, but it's like there's nothing you can do about it. So you do what the majority of people questions you are making.
Yeah. Yeah. Good technical stuff. But it's really nice material to go through. It's quite logical. Did you part of the creating process or yeah. Well done. There's a lot of work in it. I mean.
Yeah. Well, it's been across several years.
Yeah, but still, like, there's a lot of.
Yeah.
Whoa, it's quite impressive.
And people are using it a lot.
Yeah. So yeah.
Does the cycles of amplification change the shape of this diffraction?
The
number of like cycles that you do on the amplification,
right.
because it would change the number of UMI, but then I was just thinking
Yeah.
whether it makes the
Good
diffraction
question.
point
Yeah.
nicer.
I wouldn't expect it to, but there's a good chance it might, due to some weird biases, because sometimes you have. Yeah. Unless you have like a weird bias, it shouldn't.
Okay.
So because you would expect that all
Would be amplified the same.
the same,
sort of
yeah,
even the noise.
yeah. So then the whole thing should just be elevated, but not changing its shape. But if you have certain cells that have, like, for some reason, a higher GC content, they might have more amplification to it. So some cells might be amplified more than others for some reason. I don't know.
Okay.
If that happens, then that's gonna be changed. Yeah.
And the other question is there any way you can map to like a exon-exon junction?
Right. That's what star does, basically. So you mean if you have a
Because you were talking about it before, and you said that sometimes some isoforms share the same exon, and then you can't really tell whether this read is coming from isoform 1 or
Oh, that's
isoform 2.
coming back to the isoform.
Yeah.
Yeah.
And then if you actually try to kind of map to exon-exon junction,
Yeah.
then you can tell that this is isoform.
Yes. That's how you tell apart isoforms.
Yeah.
Yeah.
But is this something that you can actually do?
Yeah.
With star as well.
Yeah. Well, star is actually designed to map junction reads. So if you have a read that spans an intron, it will find that. That's so-called junction read. Is that what you mean?
Yeah, but that's easy when you are aligning to the whole gene.
Yeah.
The question when you start doing the transcript analysis.
Right.
So that was my point, because
So usually you don't use star for that sort of thing, because
Okay.
usually star you use for genome mapping, because at the point of star is to be able to tell apart introns, and they are being
sort of like differential expression between entire genes and not like specific isoforms.
yeah, for isoform, I probably wouldn't use star.
So what type of things do you use?
So I think Salmon, like one of those, the pseudo aligners, they claim themselves to be able to do that sort of thing. I don't quite buy it, but I think that was the last time I looked into that was like 3 or 4 years ago, and there were some weird things, but they claim they can handle that sort of thing.
Okay.
So things like Callisto and Salmon.
Okay.
So in the section of pseudo-aligners, if you read around the software, I think their big thing is to be able to do isoform.
Okay.
But I always find it like a bit tricky. But you might be lucky to do that. Yeah. So yeah. But isoforms are tricky. Yeah. Cool. It's getting coffee. Sorry.
They were quite alive.
Yeah, for my own mark. That's quite geeky, is it?
What's on the, there's a parrot.
Yeah, I know.
There's
It's
a
a
drone.
little weight. Can't do mathematics. But it's a bit more exciting than those guys. So at least you never, like, if you misplace your cup, then you never know which one's yours. This one.
Yeah. It's quite distinctive. Yeah.
It is. It is
If
a
someone
bit.
steals your mug.
Yeah,
you
yeah.
know.
I will know. I will know. Track them down. Right.
Yeah, because I was like, don't understand.
Ask a question. What do we do time-wise? So should I be a bit more quicker to go through 7.1 because 7.2 is 7.3 is visual stage, and that's something we should do before lunch.
Definitely try to start this by
Yeah.
lunch.
By lunch, yeah.
Yeah, 'cause
Yeah.
that will take a long time.
For them to.
Yeah, because they're getting used to our
Yeah. Yeah.
Try and start that.
Yep.
Then they can go to lunch to work on that. Back again.
Yeah.
Again, work on that and have that.
Great. Okay. And yeah. So basically, I'll just talk through, and then they do it until on time. And to lunch is 12:30. What do you do?
Anytime 12 and 1,
Okay.
we can stop.
Yeah. We
Get to a good point.
Yeah. And we say like 1 hour lunch time, or
Yeah, it's 1 hour
Okay.
lunch.
So wherever like we happen to stop.
Yeah.
Okay. So would you think once they do their own stuff, like you give them like another 15-20 minutes to start, maybe, and then say, okay, just continue after lunch. Yeah. Okay. Brilliant. Good. Thank you very much. Yeah. On probably that's for after the course, but some feedback on the structure of the thing. I think there's some. I would have some minor improvements if there.
Yeah, no.
Maybe we can, I guess, talk about that after the course or during the course, or and I don't know when.
Usually we make a bunch of notes at the end of the course.
Yeah.
Either during the course or at the end of the course, and then afterwards figure out
Yeah.
what we're going to act on.
Yeah. Should I put those notes in Etherpad?
No.
No. Okay. Good.
But
I'll just write them down, and then
yeah,
we.
if notebook or I can use paper and you can just write them down
Yeah.
as you go.
Yeah. Cool. Brilliant. Thank you very much.
Also, if anyone asks a question that we don't have answered on there, write it down too. We try
Okay.
and
Yeah.
whenever
Yeah.
we get a
Improve the
question
material.
coming up to improve the material by.
Cool. Just keeping it all in my mind. Brilliant. Okay. I think I'll carry on. Brilliant. Oh, there's some issue with the coffee machine. Just waiting for them to get their coffee. Yeah, no, that's problem with the coffee machine. I'll just double check because I want to continue it. I'm waiting for. Oh. Okay, we just start after you got your copy. Okay, continue in a minute. They just need to get a coffee. So. Yeah, they're still because there's a bit they just refill at bad timing. I'll just wait for those two guys and then they'll. Can you say how you pronounce your name properly? Rocio. Just Rocio. Is that? Yeah, Rocio. I can't do the Rocio. Okay. Good. That's good. They've worked out Etherpad. Have we? So they know how to handle that.
Well, all of these are questions.
Ah, brilliant. That's amazing. Cool.
Some of them are.
Yeah. Okay, I'll just remind them again.
You see 39 people in.
Wow.
Yeah.
Okay, brilliant. Okay, guys, listen up. We'll just continue with our exercise until the lunch break, which is just about an hour's time. And just one more reminder, just in case you haven't worked it out, I think a lot of people have. There's this Etherpad, which is if you click on that, which is in the tab on the bottom of Firefox, takes you to a shared document. You can ask questions, and our demonstrators are trying to answer them in real time. And I think there's also people on the internet that are participating. So yeah, so have a look at those ones and don't get trolled. Okay.
So anonymous, so if you ask a stupid question, no one will know. So happy to answer.
Oh, we'll see them typing. Good. Okay. So we just continue on from the first filtering step. So that's a really essential filter step in single cell to separate the real cells from the background noise. You don't want to have the background noise. They don't mean anything. And yeah, so this histogram is basically yet another way to what I was just telling you about to separate that. And I think the other way I showed you before the break is probably a better way to work out that cutoff point. In this specific example, we just use the cutoff point where the red line is of arbitrarily 25,000. So every barcode that has less than 25,000 reads assigned to them, we will just designate as background noise and exclude. How many are those for that data set? Well, we can use normal R. So if you know R, then that helps you a lot to do research, because a lot of the software is fiddling around and trying to play around, and having a good understanding in R is usually good. So in this particular case, you can just use the table command, which is just this box standard R, to work that out. And this is like a normal filter step, so that unequal, like greater than sign, basically tells you filter everything out that is, keep everything that's higher than that number. It's just really normal R. So if you actually do that, the table command will tell you how many are been filtered, and that's 46. And the result of that filtering is in this vector here. So that's a vector of 800 something, whatever like the number of stems cells were in our data set, and each of them has a true or false. Maybe I'll actually show you that. Just filter filter by total counts. If you just execute it, that's 860 something entries with true or false, and true if it's above 25,000 and false if it's not. Okay, so that's a vector we're going to use later on, so that's why I might keep that in your mind. And another filter is based on detecting genes. So again, we are in cleaning our expression matrix. We want to get rid of both the cells that we don't want as well as genes that we don't want to look at. And here we are looking at genes at both at the same time. So we want to exclude the cells that don't have a minimum number of genes expressed. Kind of the same as reads, but using genes as a count. So that's the same thing. We can look at the distribution of how many genes are expressed in each cell. So that's usually what people care about. How many genes do I hit in my cells? And here the answer is between 8,000 and 10,000 on average. Each cell has between 8 and 10,000 cells expressed. Usually only 1 or 2 counts per gene, but that's the amount. And so here we can again set a filter of cells that have less than, say, 7,000 genes expressed. Okay, so that's this red line. Also, you first? The question is about doublets. So if you have doublets, the doublets are if you have 2 cells that are for some reason encapsulated in a droplet, so you have double the amount. Yeah, okay. So usually there's no good way to identify them because you're using the barcode. They both have the same barcode, and usually you would get them by they usually have more expression. So if they have a lot of reads them, they could be potentially doublets, but it's really not a good metric to tell. The only way to really tell is if you run a mixed species experiment. If you have cells from different species, you can find out doublets. If half of the cells map to one species and the other half of the reads map to another species, then you can identify them. But other than that, there's no really good way to work it out. Is that answering your question? Yeah. There was another question on. Yeah. So yeah. Yeah. So I think this one is probably not the best filter. Yeah, that's just one filter, and yeah, you sort of have to play around. A bit of biological knowledge needs to feed into that, and you have to sometimes run this analysis a couple of times and change the parameters a bit. So I think the more reliable one is the filter based on the count itself, using that inflection point. I think that's usually what people use. Yeah, but there's more filters than that I'll show you. So they're all based on experience in a way, and it's kind of early days to work out the thresholds. Okay, so how many cells did we filter out using that threshold? 116. Some of them will be the same as the one we would have filtered out using just the counts early on in the chunk. Okay. So that's a filter step you can use, and again, that's a bit arbitrary. And another telltale signal I told you about earlier is the ERCC and mitochondrial genes. So remember, we've fed the information on which genes correspond to the ERC probes as well as mitochondrial genes. Now is the time to take advantage of them, and the way you do that is using, first of all, plotting what the deal is with them. How are they distributed across your cells, and best explained with a picture, as they say. Okay, so if you get that figure up, that basically tells you for each individual cell in the experiment corresponds to one of those dots, and the color of the dot corresponds to one of those 9 batches that we were talking about. So, okay, but more importantly, the x-axis has the number of features. So that's genes. So that's the gene expression. that corresponds to the 8 to 10,000 genes that are expressed per gene. And on the y-axis, importantly, that's the percentage mitochondria. So how many reads are actually derived from mitochondria per cell? So you see 5, 10, 15%, most of them are around 5%. So 5% of the cells have mitochondria expressed as reads, okay? This section. And some of them are even above 10%, and that's a sort of threshold you can use to exclude. So you could argue this is dead cells because they have a high threshold of mitochondria. And using that plot, you can tell. Because sometimes you have a population by itself that are just enriched in high mitochondrial cells. Okay, and again, those plots, you should always do the sort of plots if you filter to get a sense of your data. And if you, for example, came to the conclusion that 10% is a good threshold, then you can use that. So that's sort of figure that tells you that. Usually 10% is what people use. And you can do the same with ERCC probes. Where's that? That's here. Okay. Yeah. So again, that's the number of genes on the x-axis and the percentage of counts that are ERCC probes here. And this one actually gives you two populations, and you could argue that's all dead cells. But interestingly, they all belong to the same batch. So that's something where you just probably should not have like a plain filter. You should probably have a closer look on what the experiment actually was designed to look like. And in this particular instance, it turned out this batch had the cells in that batch are just smaller. And if you have smaller cells and you inject the same amount of RNA, the relative amount of RNA is higher because you get less RNA in smaller cells. That's why the percentage of ERCC high on those cells on average, and that's why they are in a separate population. In that particular case, you could argue that you should have two different thresholds, one for smaller cells and one for bigger cells. Okay, and that's different for every experiment. You might have a widely different experiment with different batch effects and sizes and weird conditions, and you have to look at those and carefully think what's going on. Can I explain that trend with my biological knowledge? There's no flat-out solution for that one, but in this case, it turns out it's the size of the cell, and you shouldn't just, because if you just filled out 5%, you would lose this whole population, even though I think those ones are legit. Okay. We do it anyway, but not as strict. So, okay. Again, setting the threshold for mitochondria to 10%, and excluding that batch, we filter out 96. So we actually, in this particular case, we actually exclude that batch because it's weird and small. And then I guess you could analyze it separately. But for the remaining ones, we're just setting a threshold for mitochondria to 10% as another filter. Okay. And so we've done a series of 4 filters at this point, and we can ask the question, how many cells have we filtered out in total based on those 4 filters? And you can, again, R is your friend, so all of those are give you 4 different vectors with truths and false. You can ask the questions, how many of them at least don't match any of those filters, so how many are good cells, and you can connect it up with this ampersand, and that tells you number of genes that are common to all filters, and table again is your friend, and yeah, so it's 207 will be filtered out because they don't pass one of those 4 filters. And there's a lot of them that are shared, so they match 2 or more filters. Okay. Any questions on that point, just feel free to ask. Good. Okay, so that's filtering. Filtering manually. There's also some software that claim they can do that automatically, so you don't have to worry about any of those thresholds. They just try to work it out by themselves. And I'll just bring up this guy here. that's just nicer to have it here. Yeah, automatic. So in Skater, that's just the package we're using. There's an automatic filter which is based on those. They've worked out those 6 features are telltale signals to tell apart the cells that you want to filter out to the ones that you want to keep. And so that's kind of inbuilt. And if you run, you can stick those 6 features into a PCA. So you take all the cells and all this expression matrix, but as columns instead of the counts, you use this 6 filters or features, and you stick it into a PCA. So that's basically munching it all together and trying to work out some sort of groupings and cells that are sort of standing out from other cells. I'll just show you a figure. So that's, oh, it didn't produce anything. Okay, I'll just show you here. So that's what should be produced. So that's PCA. That's basically showing you which cells are close to each other versus others. So there's a bit of an outlier group here. You could argue that's probably weird cells. You should filter them out because they are different from the bulk of the cells. And indeed, the automatic filtering is indicated in orange. That's what this program magically filtered out to be, worked out to be filtered cells. So that's the orange ones. And the ones that are manually, that we manually specified as outliers are the ones in circles, whereas the ones that we keep manually are the triangular ones. So basically, the orange ones are a subset of the circularized ones. So it's basically the same ones, but we had more. We worked out there's more to be filtered out than the automatic filtering, and you can actually compare those 2 numbers. And so you basically overlap that orange with the circles, and you work out that we filtered out around, I don't know, 200, and the automatic filtering 73, and which is included in those 200. So we filtered out more manually. Okay, that's filtering, which again is a bit on the boring side, but it's really important because if you don't filter properly, you do not get good results. And the last filter before I leave you to a practical is based on genes. So for the time being, we only looked at gene numbers and this sort of thing. But you can also look at which genes itself are expressed for each cell. So you can basically rank genes. So that's a nice figure. So you can rank genes by their abundance. So what gene is expressed most in all cells, and what's the second one and the third one? And usually a good sort of plot is to plot the first 50 genes. So that's genes on the as rows, and I'll just that's on the y-axis. You got the percentage of total counts. So, and each of those lines corresponds to what that gene is expressed in each individual cell. So we got about 800 cells, so we got 800 of those lines, and each of them shows the expression level of that gene in that cell. You see, the first most expressed gene is about, I think it was about 1%, and the second one is just a bit lower than that, and that's kind of like a nice distribution. That's something you expect. You always have like a most expressed gene. Sometimes it's RNA, sometimes it's something else, but usually that's the sort of thing you want to look at.